in front of you about the role of stromal heterogeneity in immunosuppression and immunotherapy resistance in cancer. This is great to be able to be with you despite the sanitary crisis. I really appreciate to be able still to speak about science. This is really important for all of us, I think. And these are my disclosures. Do you see the second slide?
Yes, everything is okay.
Okay, fine. Thank you. And just before I begin, just would like to introduce myself, to discuss with you about CAF. I just would like to say what we have done before and why we have been interested by carcinoma-associated fibroblasts. And in the past, we have been interested by the pathophysiological consequences of a chronic oxidative stress, meaning a stress which is faint enough to remain totally compatible with cell survival. And what we have seen in cancer is that there is reactive oxygen species promote the accumulation of a hypoxia-inducible factor even under normoxic conditions. And this leads, and this converts the fibroblast into myofibroblast. And this increases stromal heterogeneity and increase tumor metastatic spread. But in addition to that, if we found that reactive oxygen species increase tumor development and metastatic spread, what we found also is that in the meantime, it can improve response to treatment. And this we observe at first in ovarian tumors, where we established the link between reactive oxygen species and the MIR200 family members, and we found that the oxidative stress related patients with an oxidative stress signature survive much better than the fibrotic one. In addition to that, we found exactly the same thing in triple-negative breast cancer, where we found that indeed oxidative stress signatures can be associated with improved survival, and we established two different mechanisms, one which is linked to autophagy and the YAP signaling pathway, and the other one to DNA repair. Even more recently, we identified OXPHOS and metabolic heterogeneity in ovarian tumors again, linking this oxidative signature and respiration capacities to the promyelocytic leukemia factors and ROS accumulation to chemosensitivity. But then, in addition to all of that, what we found is that we also established recently some stromal heterogeneity in breast and ovarian tumors. And we identified different CAF subsets, and one of them was associated with accumulation of regulatory T cells. Even more recently, we found that these CAF subsets involved in immunosuppression, together with another CAF subset, is associated with huge increase in metastatic spread specifically. And finally, as this CAFS1, what we call CAFS1, is associated with immunosuppression and metastatic spread, we wonder whether these CAFS1 cells could be by themselves heterogeneous. And then we analyze this question by single cell analysis, and we indeed identify different clusters of CAFS1 cells. And then today I just would like to really focus my seminar on this carcinoma-associated fibroblast heterogeneity. Just to rapidly come back on what we have found in the past, what we established is that we identified 4 carcinoma-associated fibroblast subsets that we called S1, S2, S3, and S4 in breast and ovarian cancer. Just to be really fast, the CAFS1, which are FAP positive and CD29 male, and the CAFS4, which are CD29 high, are strictly detected in tumors. They are both SMA positive myofibroblastic cells. CAFS1 are more characterized by extracellular matrix and inflammatory signaling, and CAFS4 are more associated with vascular signature and oxidative metabolisms and respiration metabolisms. In contrast, the CAFS2 and CAFS3, which are found in tumors as well and can be defined as a CAF, are also found in healthy tissues and are more reminiscent of resident normal fibroblasts. I will not enter into all the details because this has been published 2 years ago, but we found that the Fab-positive CAFES1 cells are really specifically associated with immunosuppression, meaning with a high content in FOXP3 regulatory T cells and reduced content in CD8 positive cytotoxic T lymphocytes, while in contrast, a triple-negative breast cancer enriched in CAFES4, for instance, exhibit lower proportion of FOXP3 T lymphocytes and higher proportion of CD8 when we compared with a triple-negative breast cancer enriched in CAFES1. And what we also identified is that this immunosuppressive function of CAFES1 is associated with a multi-step mechanisms. The CAFES1 cells are able to attract the CD4 positive, CD25 positive T lymphocytes by secreting high level of 6L12. And 6L12 is also able to promote the survival of these T cells. We found also that the CAFES1 cells are really able to retain the T lymphocytes at their surface through PDL2, OX4 T ligand, and JAM2, which are molecules highly expressed by CAFES1. And finally, we found that the CAFES1 cells are also able to promote the activation of Tregs, meaning CD25 positive, FOXP3 positive T cells, and these cells are really able to reduce the proliferation of T helper cells, and this is associated to huge expression by CAFES1 of DPP4, B7H3, and CD73 in CAFES1 cells. In addition to this function, this immunosuppressive function, what we also demonstrate recently is that the CAFES1 cells are able to also interact with CAFES4 cells to promote metastasis in breast tumors. And we demonstrate that in two recent papers. And the mechanisms by which these two cell types promote metastatic spread are different. The CAFES1 cells promote metastasis through 6L12 and TGF-beta release, which directly promote and or initiate epithelial to mesenchymal transition of cancer cells, while the CAFES4 exhibit more metastatic activity by acting on the extracellular matrix remodeling. Thus, we observe that there is different roles mediated by CAFES1. CAFES1 cells, and these Fab-positive CD29 MED cells, SMA-positive cells, are really able to promote immunosuppression and metastatic spread. And we wonder whether by themselves it could be heterogeneous. And we first highlight that by performing cytospectral analysis on breast cancer tumors using 6 different CAF markers. And when we analyze, when we observed when these cytospectral analysis create trees and promote among hierarchies of cells of related phenotypes. And what you can see on this tree is that you have 4 main branches, confirming the existence of the 4 main CAF subsets. But in addition to that, what you can see is that then, for instance, the CAFES1 cells are positive for FAB, the CAFES1 and CAFES4 are positive for SMA. But what you can clearly see is that indeed, in these CAFES1 cells, you can have different expression of the SMA protein here in these cells, suggesting that indeed these cells could be by themselves heterogeneous. And we try to address this question by performing single cell data. And indeed, we found 8 different Fab-positive CAFES1 clusters in breast tumors, and we analyzed more than 18,000 CAFES1 fibroblasts isolated from 7 breast cancer patients. And as I said, we identified 8 different clusters here highlighted by this UMAP analysis and differential expression of the different genes characterizing each of these cells. We first observed that these different clusters can be divided into two main groups. First one gathering 3 clusters, which are really which express the ICAF signature previously identified by Daniel Ohlund in 2000's lab. And the MyCAF signature, expressing more myofibroblastic signature, exhibit huge expression in the, let's say, the right part of a UMAP representation. And then we decided to call these different CAF subsets according first to the ICAF and MyCAF signature enrichment and to the different signaling pathways that we could identify based on the differentially expressed genes. And what we found is, for instance, in the ICAF clusters, we identified the interleukin ICAF, which is quite reminiscent to certainly what Florian showed before. The detox ICAF, which are more related to detoxification pathway, and the interferon gamma ICAF, which is obviously characterized by an interferon gamma signaling pathway. And in the MyCAF subsets, we identified an extracellular matrix MyCAF cluster, a wound healing MyCAF, a TGF-beta MyCAF, an interferon alpha-beta MyCAF. And these different clusters are really characterized by specific signatures. And all these clusters, in fact, are found in all patients, but in different proportions. This is the cluster distribution that we can highlight it. Each color represents the same pathways, the same clusters as here. Then in this cluster distribution, in each of the 7 patients analyzed by single-cell analysis, we can clearly observe when we analyze the 5 most abundant clusters, which represent 91% of the total cells, what we can clearly see is that we can detect all these clusters in all patients, but in different proportions. And we wanted to validate this observation by using another techniques. And to do so, we wanted to validate that by multicolor flow cytometry. First, we tried to identify specific markers of these different clusters. And here are the representation of the different markers that we used. These are the representation of the mRNA expression level of each of these markers that we use to analyze these different clusters by flow cytometry analysis. And we established a gating strategy. We first, by flow cytometry, we first gate on the CAFS1 cells and FAP-positive CD29 MERS. After that, we differentiate the anthrax-positive cells, which differentiate between the ICAF and the MyCAF cells. Among the MyCAF cells, we also differentiate with SDC1 and LAM5, and after with CD9. And for the anthrax-negative ICAF, we differentiate the two cluster 1 and 2 by GPC3 and DLK1. And then what we found by using these analyses and these different markers by multicolor flow cytometry, we found exactly the same thing as by single cell, meaning we have been able to identify these different clusters in the different patients that we analyzed, but in different proportions. Thus, we next analyze, okay, what could be the consequences of the presence of these different CAF subsets in the patient? What we found is that as we have identified that the CAFS1 cells are really associated with immunosuppressive environment, we wonder whether these different CAF subsets, CAFS1 subsets, could be associated with different immunosuppressive functions. And then we ask that question also by multicolor flow cytometry, where we analyze the composition of the lymphocytes. And what we found is that indeed For instance, the CAFES1 cluster 0, ECM-MyCAF, is really anti-correlated with cytotoxic CD8 T cells and NK cells, and correlated with CD4 that are positive for CTLA4, PD1, or TIGIT. In addition, again, we found that the CAFES1 cluster 3 is also associated with an immunosuppressive environment, it's called TGF-beta-MyCAF, and this cluster is anti-correlated with CD8, correlated with CTLA4 positive, CD4 positive T lymphocytes, and correlated as well with macrophages, CD14 positive macrophages. And finally, we found that the so-called wound MyCAF cluster, cluster 4, is globally not associated with immunosuppressive environment, but globally with a huge immuno-related phenotype, meaning with a huge infiltration of T cells. Okay, based on that, could we identify a link between these different proportion of clusters and the immunotherapy response, as we have identified the link of some of the clusters with accumulation of CTLA4 positive or PD1 positive T cells? Then we try to ask these questions first by using metastatic melanoma cohort of patients established by Roger Lowe, in which in this cohort, composed by metastatic melanoma patients, these patients have been treated by pembrolizumab, and this study established 15 responding patients and 13 non-responding patients. And then we analyzed the impact of the global CAFES1 and CAFES4 signature. And what we found is that clearly on this gene set enrichment analysis, we found that the non-responder patients are really enriched in genes expressed in the CAFES1 signature, while the genes composing the CAFES4 signature is totally found both in non-responders and responders. Then what about the CAFES1 clusters? We found that the CAFES1 clusters, the cluster 0, ECM-MyCAF, the cluster 3, TGF-beta-MyCAF, and the cluster 4, wound-MyCAF, then these clusters exhibit a huge, their signature exhibit a huge overexpression in the non-responders patients compared to the responders. In contrast, the cluster 1, 2, and 5, meaning the ICAF clusters, are not associated with are not indicative of immunotherapy response. And to try to confirm these results, we have also established in collaboration with Gerard Zalkman at Bichat Hospital, we have also established a cohort of non-small cell lung cancer treated by immunotherapy, and we had access to 79 samples embedded in paraffin, and we extracted RNA from 56 adenocarcinoma and 23 squamous cell carcinoma. You can clearly see that in these patients who have been able to respond to immunotherapy, of course, the responders survive better than the non-responders, and this is more or less the same thing whatever the subtype of non-small cell lung cancer that we had access to. And we also analyzed the impact of the CAFES1 and CAFES4 signature in this non-small cell lung cancer. And what we found is that again, the non-responders the CAFES1 signature is higher in the non-responders compared to the responders, while in fact the CAFES4 could be even in the reverse, perhaps higher in the responders. And here again, we analyzed the different clusters, and the cluster 0, cluster 3, and cluster 4 found associated with immunosuppression environment. Then this cluster, ECM, TGF-beta, and wound, are associated with higher expression in non-responders compared to responders, and then indicative of non-response to immunotherapy, while the cluster 1, 2, and 5 are not. But I hope I convince you that indeed there are some CAF heterogeny in breast as well as in other tumors. Just would like to mention that we identified these different clusters not only in breast and ovarian, but also in non-small cell lung cancer and in, for instance, in head and neck cancer. This is clear that these different subsets exist in different tumor types. Then we identified these 4 CAF subsets by combining different markers. We established that the CAFS1 Fab-positive cells exhibit a key role in immunosuppressive environment. That both CAFS1 and CAFS4 and Fab low or negative CD29 high cells are really able to promote metastasis through complementary mechanisms. And we found also that the CAFS1 cell by themselves exhibit intra-subset, we can see intra-subset heterogeneity by identifying different clusters that accumulate differentially in patients. And we found that 3 of these clusters exhibit an immunosuppressive role, and they are involved in resistance to immunotherapies. In particular, this has been associated in melanoma and in non-small cell lung cancer patients. I just would like to thank the members of the team who perform all these experiments. First, Anna Costa, who performed the work on immunosuppressive environment. Rashid Hossin, who performed the analysis of the immunotherapy resistance and CAFS1 subset, together with Jan Kiefer, who is a bioinformatician in the lab, who has been involved in all the projects that I showed you. Brigitte Bourachot also worked a lot on all these different aspects. Florian Pelon also has been working on metastatic spread, and together with also Ilaria Magagna. Just would like also to mention Charles Bernard, who helped us a lot in bioinformatic analysis, and 2 permanent researchers in the lab, Virginie Miolet and Géraldine Gentric. And of course, I really like to thank all our collaborators, our funding providers, and patients and their families for their participation in our study. Thank you very much for your attention.
Thank you, Fatima, for this nice presentation. You know, it summarizes years of work. You put it in 30 minutes. So when you start talking about CAF sub 1, 2, and 3, and 4, I said maybe, okay, Fatima, now she has the answer. There is only 4 subtype, and then we can regulate their level, and then we can make stratification of the patient. And then the situation is very complicated with this cluster. So at least you have 5 cluster, 1 in subtype 1. So I guess you have the same situation in the other subtype. So in the end, we'll have like how many cluster? You have like 30 cluster. How are we going to deal with this? So how are we going to manage patient? The important task is to choose patient that having this kind of cluster and in order to give them specific therapy.
Absolutely, yeah, this is exactly the point. And for your first notification, I think that indeed, yeah, we are really dealing with the emerging part of the iceberg. In fact, it was exactly as if we were wondering whether we could deal only with CD3 without considering CD8 or CD4, or even now the different subsets of CD8 and CD4, for instance. Then we are really dealing with CAF exactly in the exact same situation. Indeed, CAF are really as heterogeneous as we have initially observed for immune cells, and we have really to understand what is going on with CAF. And indeed, as you said, when you have accumulation, or let's say, of less activated fibroblasts, such as CAFS3, for instance, which correspond more to resident-like fibroblasts. In fact, these, for instance, accumulate in luminal breast cancer patients compared to HA2 and triple-negative breast cancer patients who accumulate more activated CAFS1 and CAFS4. And in CAFS1, we have observed different accumulation of the different CAFS1 clusters. And as you said, when you have a triple-negative breast cancer patients with some specific CAFS1 clusters, most probably they will not respond properly to immunotherapies. Then indeed, we have to deal with that. Absolutely.
Yeah, actually, this is what Julie Panka is asking. She's asking, I mean, in respect to what we just said, can you then imagine to stratify patient thanks to CAF S1 signature before treating them? Feasible? Yeah. Can it be applied to colon cancer?
Then for colon cancer, we are working in close collaboration with Arnie Ostman in Sweden, who is performing exactly this type of analysis. And indeed, yes, we have been able to identify, in collaboration with Arnie, we have been able to identify the different subsets, yes.
Yeah, and you have a question now from Jean-Philippe Girard. Maybe just go ahead. You have access to the. Okay. Yes, go ahead, Jean-Philippe.
Hi, Fatima. Fantastic talk. This is very interesting. The subset, the CAF S1 subset. I have a question about the CAF S4 subset. You say that in triple negative breast cancer, they are associated with CD8 T cell. Therefore, this is good or not? And do you think there are also different subsets of CAF S4? Good subset, bad subset, like the S1?
This is a very interesting question. Yes, in triple negative breast cancer patients, we have found that in contrast to CAF-S1, the CAF-S4 are more associated with a reduction of FOXP3 and an increase in CD8. And this is interesting because we perform single cell analysis on CAF-S4 as well, and we identified a bit less number of clusters. We identified 4 different CAF-S4 subsets, and we are trying to understand what is going on behind that. And we found signatures, again, some extracellular matrix related, some of those interferon related. Then we are trying to understand where they are acting and how they are acting on metastatic spread.
Okay. Thank you.
And we have a question from Marie Caneo. She's asking any idea of the mechanism that dictates the establishment of CAF subtype populations. Do we know why?
This is a very good question as well. It's not totally clear. What is clearly in mind is that obviously cancer cells can modulate the resident fibroblasts to promote and drive CAF-S1 differentiation. This is something which is in the air, let's say. Then indeed, that the proximity of cancer cells, in fact, the CAF-S1 cell is really close to cancer cells. When you can have, for instance, in a luminal patient where you do not have a huge number of CAF-S1, the CAF-S1 are really detected in close proximity with tumor cells, suggesting that indeed there is a reciprocal crosstalk between cancer cells and CAF-S1. And the hypothesis is that the resident fibroblast in the tumors, CAF-S3 for instance, can be modulated by cancer cells and induce for CAF-S1 differentiation. And for the CAF-S4, as they exhibit a pericyte signature, people think that they could derive from pericytes. Then really different origins from CAF-S1 and CAF-S4, these 2 SMA positive type of fibroblasts that could be derived from 2 different sources.
Okay. And we have Pierre Coderi is stating, superb talk, many thanks. Do we know if all of these CAF subtypes originate from a common ancestor? Can we switch phenotype from one to the other?
Okay, yeah, this is also a very good question because it's more or less related with the previous one. But if we speak only about the CAF-S1 clusters, yes, indeed, Pierre, we think that indeed they are coming from one specific ancestor, and exactly what we are doing now, we are establishing by bioinformatics and biological experiments we are performing. We think that indeed one cluster can give rise to the others, and we are trying to identify the mechanisms involved in this plasticity. But yes, we think that there is one ancestor that can give rise to the others. For the CAF-S1 clusters.
Okay. And Corinne Bosfish is thanking you about this great talk, and she's asking, how do we think your result about CAF-1 can be translated to improve treatment, especially to immunotherapy?
Yeah, here again, this is a very good point. What we think is that we think that we can't then we could stratify first patients for immunotherapies, not giving immunotherapies to patients with high CAFS1 content. Okay, this is one of the first points that we can do. But of course, we would like to treat them to be able. Then we need to target the CAFS1 cells. And then we are trying to deal with that by performing some targeting of CAFS1 in addition to the immunotherapies, and just trying to see if we could, by targeting CAFS1, increase the immunotherapy efficiency.
And in the same orientation, kind of question, Bérangère Pouladabalat, she's saying, Salut Fatima, do you think it would be possible to revert CAF subtype to improve treatment response? I think it's the same.
Yeah, thanks Bérangère for asking this question. That's exactly, as Majid said, that's also associated with the previous answer. But what is interesting in your question is that indeed you don't say, okay, we would like to kill the cell, but we would like to revert them. And this is really interesting because I think that's the way to do. The problem that we have really to analyze and to identify the plasticity of these cells and the mechanisms involved in this plasticity to be able to revert them. And it will be easier for the moment to kill them, but I do agree that we would like to revert them. But that's exactly what we are trying to understand to be able to do so in patients.
I think you answered the question to Julie Panquet. She's asking, have you identified in a treatment that could change this heterogeneity in patients? Means that you can switch from one to the other one.
That's exactly what I said. That's exactly what we would like to do, precisely, yes. But for the moment, we are far from that, clearly.
Okay. So thank you very much, Fatima, for this nice presentation. And also I will take this opportunity to have two messages to deliver. The first one, the previous session are already saved to record for the patient that missed the previous session. There you can go just on the platform and then you can listen again all the previous speaker. And also don't forget, tomorrow we have another session about RNA and cancer. It will be also impressive like this one. So I will take all the speakers. 4 of them, it was a great talk. We learned a lot, and you can see it's very difficult to manage tumor interaction with their microenvironment, but at least we have some ideas, and also you are welcome to interact and also to improve all this kind of science. Thank you very much, and hopefully to see you physically in the future. Thanks.
Thank you. Bye-bye.
Thank you. Thank you. And take care.